All right, everyone, welcome to the final day of this workshop. Thanks for joining us today. Want to jump and hand it over to Orr and Sergio, who will be giving you your next lecture. So Orr and Sergio, over to you.
Great, thanks, Christine. So good morning, everyone. I'm Orr, and me and Sergio are going to kind of guide you through the last day of material today, and it's going to be really some interesting kind of concepts where we're going to focus on identifying cell subsets from the single cell transcriptomic data and try to interpret what those subsets mean. So let me share my screen. Okay. So feel free to interrupt with any questions over the Zoom chat, or just raise your voice, or use Slack as well. Okay. So, through this workshop and through your prior knowledge, you know that there's just this really huge diversity in cell types, states, and interactions across human tissues. And here's an example of that, where we have microscopy images of the skin, the brain, the blood, and the small intestine. For instance, in the blood, you can see monocytes migrating into the tissue and differentiating into macrophages. In the small intestine, you can see these very differentiated epithelial cells that line the crypts. And then we also have disease tissue like liver cirrhosis or breast cancer, where there's this change in the tissue structure and the cell types and states that are present. And so we need computational methods to really identify these cell types and states once we have made these sort of high throughput transcriptomic measurements. So Daniel yesterday showed you kind of the first part of this, the computational part of this, which is the pre-processing, where you did your single cell RNA sequencing experiment, and you took it off the sequencer and did alignment of reads and generated a gene expression matrix. So you quantified for every cell what genes are expressed and which cells are even present in your data. Then he showed you quality control, where you filter low quality cells out and remove genes that aren't well measured. And then finally, he showed you normalization of the data, which we often do to correct for differences in sequencing depth across different experiments. So what Sergio and I are going to show you are the next steps, which are kind of clustering the data and biological interpretation. So start with clustering. There's multiple steps, and the first few steps involve reducing the dimensionality of the data. So each of the individual cells in the single cell transcriptomic experiment has 20,000 measurements, one for each gene. However, many of the genes are lowly expressed, or because of technical reasons, the data is very sparse, meaning there's a lot of zeros, so we don't capture the gene's expression. And then furthermore, the genes are co-regulated, which we'll discuss in a second. But what we do in this first step is we try to reduce the dimensionality of the data by just focusing on genes that are variable across the cells. And the kind of intuition behind this is that the variable genes are going to most closely reflect genes that are important to defining the different cell types and states. So if a gene is uniformly expressed across all the cells that you're looking at, it's not going to be informative in saying this is a B cell and that's an epithelial cell and that's a ciliated cell. Like, there's going to be no way for it to distinguish those because it's uniform. It's the same expression across things. Versus if a gene is highly expressed in one cell type and lowly expressed in other cell types, it's variable across your data. And that gene is going to be one that is much more likely to tell you something about the cell type or cell state. So what we can do is actually just look at the variable genes and throw out a lot of the other genes that are going to be less informative for the task of clustering the cells. So here's how we do it. We often just identify variable genes through these really simple analyses, where what we're showing here are all 20,000 genes from a single cell RNA sequencing experiment, where each gene is a dot and some of them are labeled. And we're plotting the mean expression of the gene compared to the coefficient of variation of the genes of the gene across all the cells. And we do this because there's an intrinsic relationship between the expression of the gene and its variance, meaning that the more highly expressed something is, the more variable it is. If you look at something like a Poisson distribution. So what we need to do is correct for this underlying relationship between the mean and the variance. And so we graph each gene along this sort of in this sort of plot, and genes that seem to be really off from the main bulk or trend line here, those are genes that we consider to be highly variable. And again, the reason we do it this way is we want to account for the expression of the gene and how that affects the variance. So any questions on that so far? And feel free to use, let me see if there's anything in the chat. You can use the chat or just raise your hand in the Zoom. I don't have Slack open, so actually, if there is a question through Slack, Sergio will have to tell me or somebody else. Okay, cool. So we identify the variable genes, and now from this, we go from using about 20,000 genes in the data to usually about 2,000 to 5,000 genes. Okay, so next, once we've done that, we still are in a pretty high dimension, 2,000, 3,000, 4,000. And again, this is pretty hard to work with because it's hard to calculate distances in very high dimensional space. Everything looks far apart when you're in really high dimensions. So this is like called the cursive dimensionality, which you can look up if you're interested. But there's also, beyond that, there's a lot of biological reasons that we can further decrease the dimensionality of the data. And the main one is that genes are not really independent of one another. They operate in co-regulatory modules. So you all know this, when you have a transcription factor, it will turn on not just one gene, but it will, go to its binding site throughout the genome. And if the right regulatory elements and other factors are there, it will turn on more than just one gene. It can often, a transcription factor can often turn on, hundreds of genes. So if you think of something like the interferon response in innate immunity, there's many genes. There's like dozens of genes that are all turned on together with a common sort of set of transcription factors. So the genes that are turned on, they're not really like totally independent of one another because they're all sort of turned on or induced by the same signaling event, and they work together. So what this means is that you don't necessarily need to measure all the genes because a lot of the genes are just co-varying together. So if gene A is up, then gene B is always up with it. So a way to figure this out is to use something called principal component analysis, and this will allow us to discover the gene groupings. And with principal component analysis, we go from just our 20,000 genes or our 2,000 variable genes, and we can reduce further to looking at 10 to 100 principal component scores. So principal component analysis is going to be the way we reduce the dimensionality of the data. And while we're going to just explain that, I'll just point out that there are many other methods to do this or similar things like this, including other matrix factorization approaches like non-negative matrix factorization. There's independent component analysis and multidimensional scaling, and also probabilistic methods like LDA. Okay, so how does PCA work? So let's take a really simple example where instead of having our data in 20,000 dimensions, it's just going to be in 2 dimensions. So here are cells 1, 2, 3, 4, 5 that you've measured gene X and gene Y. And what you can see immediately is that when gene X is high, gene Y is high, and when gene X is low, gene Y is low. So these are very correlated, these 2 genes. And so we can imagine that maybe they're turned on by the same transcription factor. And so every time gene X is on, gene Y is on. So ultimately, you don't really need to know the measurement of both gene X and gene Y. It's enough to know one or the other, and you can kind of infer the expression of the other. So that's kind of the intuition. And what PCA will do is it will find a direction of a direction that's kind of a combination of your genes that maximally explains the variance in the data. In that first direction that maximally explains like the variance of the genes in your data, that's called principal component 1. So we have this way to calculate the principal components, and the first one is going to be principal component 1, and everything will be mapped on the principal component 1. And so what the PCA is saying is that, you can map these points in the principal component 1, and here are their new values. They go from -4 to 4, centered around 0. And then the next principal component, so you have multiple principal components. The next principal component is going to be orthogonal to the first principal component, and it's going to capture all the remaining variation that the first principal component did not. And so what we can see with principal component 2 is that there's very little, the magnitude of those points is along principal component 2 is very small. So most points have principal component 2 close to 0. So what this is saying is that basically all your data can be just described with using just principal component 1. So you've gone from 2 dimensions using gene X and gene Y to 1 dimension where you've projected the data onto principal component 1. It's kind of this combination of effects from gene X and Y. So I'll explain that with this plot as well, which I also like. So here's a pretty similar example where we have gene X and gene Y, and these are our cells. And again, when gene X is high, gene Y is high, and when gene X is low, gene Y is low. But now the cells are a little more scattered. And what we're doing is we're rotating these 2 lines here that are perpendicular or orthogonal to one another and projecting points onto that line. And what you can see when this lines up here, going from here to here, is now when the points are projected, they are the most spread out onto that line. So keep an eye on this black line, and then once it goes from here to here, kind of going from lower left to upper right, you'll see that the points when they're projected are the most spread out along this line, meaning you're capturing the most variance of the data when you've projected things onto principal component 1, where it just kind of goes straight through the data, almost like doing a best fit line or a regression. So that's where principal component 1, you would stick it. And then the next principal component is this line that's perpendicular, and that's principal component 2. And that captures the remaining variation. So any questions on principal component analysis? Okay. And so in the real world, we don't do principal component. Oh, there's a question in the chat. Yeah, okay. GOT's question is, in terms of variance, explain what should be the threshold. So let me give context to, thanks for the question. Let me give context to that. So in the real world, we do principal component analysis on like really high dimensional data, like for what we're working with. Our gene expression data could be from like 2,000 dimensions to 20,000 dimensions. So we don't just use 2 principal components to describe the data. We often use 10 or 20 or 100 principal components. And each one of those principal components explains a certain amount of variation. So the first principal component always explains the most variation in your data. Then the second principal component explains the second highest amount of variance in the data. And the third principal component explains the third highest amount of variance in your data, and so forth. So each principal component explains less and less of the variance in your data. And ideally, right, kind of intuitively, you would think, let's try to explain as much of the variance as possible, and not kind of lose any of the variation, which links to the biology we care about. So that's the correct intuition. So GOT's question is like how many principal components to include. So there are lots of heuristic plots that one can make to see like how much variation each principal component explains, and often people will try to choose as many principal components as is needed to explain like 90 or 95% of the variance in the data. Or they will look for like an elbow in these principal component sort of variance plots. So these are all kind of heuristic things, and there's no like totally right answer, but you're also free to try a range of number of principal components. There is a wrong answer in that you never want to use too few. Like if you're using 3 principal components and that only explains like 40% of your variance, that's like not a good idea. But usually you're not going to run into troubles if you use slightly too many principal components. Good question. And there's also in the lab exercise, we go through how to make some of these decisions. Any other questions? Cool, good question, thank you. Okay, so next, now that so just take a step back. We start with 20,000 dimensional data, 1 gene for each dimension. And now we went through variable gene selection, where we have maybe 2,000 to 5,000 genes. And then when we do the principal components, we typically end up with, say, 50 principal components. So we've moved from 20,000 dimensions to this 50-dimensional space, which behaves a lot better when it comes to clustering. So what we're going to do is 2 steps in the clustering. First, we calculate distances between all pairs of cells in this lower dimensional space. So that's typically like the Euclidean distance, so similar to, what we learned in algebra class. So we do that for every pair of cells, and then we take that matrix of cell by cell distances and translate it into a graph. And then we do clustering within that graph. So we'll visualize that as follows, but again, I'll just point out there's like 100 ways to do this step, and is one better or worse? It just kind of depends on your question and application, but there are some consensus methods that people use, and we're going to talk about kind of the main method right now. Okay, so again, the goal of the clustering is, we're going to identify the different cell types and states and interpret them. Okay, so depending on how you cluster the data, you can get very different results. So here are examples from the SciPy or Scikit website, which has a lot of classical machine learning clustering approaches implemented for you. So these are the names of different methods, like you might have heard of k-means clustering or dbScan. But there's all these methods here on the columns here, and these are different data sets. And you can see, depending on the method, you'll get the clusters called in different spots. So here we're always trying to identify, 3 or 4 clusters. So for instance, in this sort of squiggle, we see that the optics method identifies an orange and a blue cluster. Affinity propagation kind of does this weird thing where the green cluster is sort of split and the blue cluster sort of split and the orange cluster sort of split. So you can see this one doesn't look like kind of correct, but these other methods look pretty good. However, the affinity propagation seems to do fine with a simpler example here with just the three sort of blobs of points. So you can see just the differences. And again, this sort of suggests that it's often a good idea to try maybe more than one clustering method, because the results you'll get will be different. And if you don't have a ground truth, which often we don't have a full ground truth, we might know some things are supposed to be there, but not all things. It might help to play around with the clustering. And in the lab today, there is a section that if you have time, you can go to and play around with different clustering parameters and see the effects. Okay, so what we actually do is we take our points that we've calculated, we take our cells that we calculate all pairwise distances. So some cells are going to be close to one another in Euclidean space, some are going to be far apart, and the intuition is that cells that are close together are going to be more likely to be from the same biological cell subset. Okay. So first, we're going to make a graph that kind of connects cells that are close by and keeps cells that are far apart. So here are cells in this sort of lower dimensional space. And what we're going to do is for every single cell, we're going to connect it to its nearest neighbors. And we choose a number like 4 or 5. So here's the first nearest neighbor to the red cell is here, and then the second is here, and then the third nearest neighbor is here, and the fourth is there. And then we're going to do that same thing for every single cell. So we'll take its 4 nearest neighbors, and we're going to connect them. And then once we've done that, we get a graph that looks, mostly like this, where certain kind of groups of cells have more connections, and certain groups of cells have fewer connections. So you can already see that there's sort of natural clusters here, like here, here, and here. Like by eye, we can kind of see that. And the idea of doing this graph is we move away from having to use these absolute distances, and we move into this sort of relative kind of sphere of doing things. And this is good because now we no longer have to worry about like magnitudes of distances and things like that, because that often is very dependent on your raw data, and that might be different across experiments. So it's often hard to kind of compare distances across different experiments. But when you move to this graph form, it's easier to compare graphs. Okay, so then we have this graph. And the last thing we're going to do with it is now for every pair of nodes in the graph or cells in the graph, we're going to ask how many of my neighbors are shared with another cell. So we can take these 2 cells, the red and this sort of cyan, and look at all their neighbors, and we ask how many of the neighbors are shared. So the red and the cyan share 2 neighbors here and here. And then we build a further graph called a shared nearest neighbors graph. And this is, again, meant to emphasize like cells that are more similar to one another from cells that are more dissimilar from one another. So the red cell here, and like, let's go back to the graph. So like this cell here and this cell over here share no nearest neighbors, but cells that are kind of in a clique or near one another often share nearest neighbors. So this is a way to kind of more quantify the similarity within the graph. So the cells are connected by an edge if any of their nearest neighbors are shared. And so we get this nearest neighbors graph, and now we do clustering on that graph. And the clustering method that we use is often, these graph-based clustering methods that are, come from, I think, kind of network and graph theory type worlds, but they're based on these algorithms for community detection. These have often been used in like social media, like Facebook or Twitter, to look at, how connected you are to your friends or followers. And, there's central people and there's people who are more distant. But regardless, this is graph-based clustering, and there's many different algorithms you'll see. Louvain and another one from another town in Europe, Leiden, are popular ones, but there's also Infomap, Walktrap, and so forth. And they're based on modularity maximization. And the idea of modularity maximization is that you want there to be the clusters to be assigned to cells that have many connections amongst themselves, but you want between clusters to have very few connections. So for instance, if this is your graph, then you can optimize the modularity by saying this is one cluster and that's another cluster. And that's the optimal, because with this choice of cluster assignments, you have lots of connections within the cluster, but minimal connections, only one between 2 clusters. Versus if you do, say, this type of approach where everything gets assigned to one cluster, yeah, you have a lot of connections within the cluster, but you also, you haven't really clustered the data much because everything's now connected. Everything's in one cluster. Versus this one here too, you can see, like, if you say this grouping here is both the green and the purple, now you have a lot of connections between things that are dissimilar. So you have 1, 2, 3 connections between 2 clusters that are kind of dissimilar. So this is an algorithm. You can look up exactly the steps, but that's how we, in the end, get clusters. Any questions on the clustering? And feel free to put a question in the chat, or just ask.
You told us that you always
I hear somebody talking, but I think you might have to turn up your volume. Let me turn up my volume too. you say, ask the question again? I think I heard a question.
Yep, that was me.
Oh yeah, I can hear you now. I turned up my volume a lot. So Daniel, what's your question? I didn't
What
hear
I'm
you.
trying to say is just you said you told us that it's ideal to use more than one clustering method.
Sure.
Can you tell us like how many you use, like the minimum, or is it dependent on the situation? Like, do
Yeah.
you try at least 2 or 3 or maybe?
Yeah, that's a good question. So I say that like it's good to try at least 2 methods. I wouldn't do much more than that because they often are different, but not like drastically different. The other thing is within a clustering, and a good question, and within a clustering method, there are parameters you can change that will change the outcome of the clustering. So you don't necessarily, it's just like trying different methods is good, but also even within the same method, you can get different results depending on some of the parameters that are set. And there's examples of this in the lab today. And we probably won't run through it, but you can run it yourself and see. And then thanks for the other question, Jelta. Sorry if hopefully I said your name sort of correct, but is the position of the cells random or what is it based on exactly? So this is actually the position of the cells in the graph is not random. Well, this is really just a depiction in 2 dimensions of the cells. In reality, we don't draw out the graph in 2 dimensions. The graph actually exists in whatever dimensionality you have after doing the principal component analysis. So usually you might have about 50 principal components. So to draw the graph accurately, where you maintain like these distances and things like that, you need to be in 50 dimensions. So this is like really like a projection of the points into 2 dimensions just for visualization's sake. But the positions aren't really random. I mean, these distances are always calculated the same way and they will always be the same. And then the k nearest neighbors, like the nearest neighbors, let's say it's looking at the 4 nearest neighbors, like that will always be the same number. So it's really the who are my neighbors that matters and who are my shared neighbors that matters. And the distances are a deterministic calculation. There are other things that are stochastic, and if you rerun them, you'll get slightly different results, but not this step. Good question. Isabella, yeah, the distances are based on, that's the distance metric is usually quantifying similarity. So the distance is like if 2 cells have very little distance, that means they're similar. And if 2 cells have very big distance, that means they're dissimilar. And the distances are just calculating this principal component space. So in principal component space, when cells are nearby one another, that means their principal component scores are similar, which means they are similar cells. And if the principal component scores are very different, that means they're very different cells. And the principal components are just reflecting different gene programs. Any other questions? Thanks for the question, Isabella. Okay. So the last part that I'll just go through before handing it off to Sergio is how do we look at differentially expressed genes? So once we've identified the cell subsets, we want to know what genes are high in one cell subset relative to another. So in bulk RNA sequencing data, you might have a gene in group, in one set of cells, say group A, and another set of cells, group C, and they form these kind of smoother distributions. This is in bulk data, but in single cell, we often have for a gene lots of zeros, and it's often referred to as zero inflated. So this means that tests that kind of rely on like really smooth distributions often aren't necessarily the most appropriate for single cell data, even though people will use them. But the main thing to keep in mind is that one thing that complicates a lot of our analysis for differential expression analysis is differential gene expression analysis is the fact that there's lots of zeros. So so to identify like what are the genes that are higher in one cell subset relative to another, there are like literally like 100 tests one could do. Here I'm showing you tests that are implemented in Seurat, which you have started to use in the last few days. So there's Wilcox and t-test, and the first is non-parametric. There's Poisson and negative binomial, which assume kind of what the distribution of the counts would look like for a given gene. So Poisson distribution, the mean and the variance are the same, and the negative binomial, the mean and the variance can be different. So you can have over dispersed data, but there's many, many tests. And so I think the next slide, yes. This is like a review article you can look at if you're interested in like all the tests that are out there. But what they did here was they tried all these different tests here, and these are different metrics for evaluating those tests across single cell data sets, with blue being good and red being poor. And so you can see these methods at the top tend to be pretty good across these metrics. Even the t-test or Wilcox do pretty well. And sometimes more complicated tests don't do so well. So here's an example of like once you've run your differentially expressed gene analysis, what that looks like. So what we're showing here is all the cells and all the genes. And here we have the clusters we identified, clusters 0 through 7. And then in the rows are genes that are differentially expressed in one cluster relative to another, with yellow being high expression and purple being low expression. So what we can see is there's a group of genes here that are yellow and highly expressed in cluster 0, and there's a group of genes here that are yellow and highly expressed in cluster 1, and so forth. And what you can see too is with any group of genes that's differentially expressed, it tends to be lowly expressed across all the other clusters. So these genes that are highly expressed in cluster 1 tend to be low in clusters 0 and 2 and 3 and so forth. So that's kind of the data we get out after the differentially expressed gene analysis. And then we can look at those genes and start to interpret what kind of cell type or state they might be defining. And then the last thing I want to show you before, like the very last thing now is like, okay, so we've identified the differentially expressed genes, and how do we visualize our data in 2 dimensions where we want to make these nice plots that we're going to show in our figures in our paper. So, or just in our general analysis. So the data, exists in this high dimensional space, as we sort of said over and over, like 20,000 dimensions. Here's just data that exists in 3 dimensions, 2 interlocking rings, or this trefoil. And there are different visualization techniques to accurately or somewhat accurately map this into 2 dimensions. So one of them is TS&E. And so here's an example of doing TS&E and projecting this data into 2 dimensions. And when you run TS&E, it might give you 2 circles or these sort of interlocking circles in 2 dimensions. Similarly, when you run it on a trefoil, sometimes you'll get this circle, or sometimes you'll get something that looks more like this. And what these algorithms are trying to do is project the data into 2 dimensions while maintaining that points that are close together in high dimension are close together in the lower dimension. So TS&E used to be the most popular approach to do this, and then now there's another approach called UMAP, which is faster and better in some ways, but both of these are acceptable ways to sort of visualize the data. And so now what you can do is take all your data and visualize it in 2 dimensions using the TS&E, and then each of these points is a cell. And we've identified the clusters through our clustering, and we've looked at the differentially expressed genes, and based on the differentially expressed genes, we've labeled what the cell subsets are. So these are immune cells from peripheral blood, mononuclear cells, and we see myeloid cells like monocytes and dendritic cells. We see lymphocytes like T cells, B cells, and NK cells, and so forth. And then within any given cell lineage or cell type, we can further divide into like naive cells and CD8 and CD4 naive cells, or different types of monocytes like CD14 positive and CD16 positive. So Sergio will go into a much more detail about like this annotation and interpretation. But just to sort of recap what we just did so far is we've gone over quality control of the data and normalization. We've gone over variable gene selection. We've discussed principal components analysis, which is one way to do dimensional reduction. And then we've done clustering by calculating cell-to-cell distances and then building graphs and doing things like Louvain or Leiden clustering. So any questions before we go to Sergio? And you'll do these steps in the lab shortly. Yeah. Okay. You can still ask questions about this, and we can answer as you get through. So I'm gonna give it over to Sergio now.
Hi everybody. I'm Sergio. I'm a postdoctoral fellow in the Schleck lab at the Broad Institute. And I will be, let me share my screen first. 1 second. I'll be continue the presentation and I will be focusing on the cell type annotation. So feel free to interrupt me if you have any questions or write in the chat. So I guess the first question that we want to try to answer is, what are we actually annotating? So as Orr mentioned before, the whole idea of this, a big part of this is we need to have cell type annotations. So we can have to annotate what groups are in these clusters to have a better idea of what we are analyzing. But the thing is, cells exist in a multiple continuum. So we are not only going to have like cell types in a stationary state. We're just not, in a lot of cases, we're going to have differentiation process. We are going to have a small transitions between small different stages. We can have inflammatory responses, antiviral responses, or even more physiological changes like epithelial to mesenchymal transitions. Which just means that in these cases, as you saw in the previous plots, there's always going to be a diffusion, a transition between one state and the other. And this is important because in general, we're going to have a tendency to try to profile these, to put cells into buckets, into specific groups, into defined populations. But it's always important to keep in mind that this is not always the case. In general, these unbiased approaches allow us to appreciate how these things change across the spectrum, because we're not defining specific markers, but we are allowed to have things that change as well in a continuum, like a lot of genes. We are going to have the ability to discover all of these differentiation process or continuous spectrum processes. Although it's kind of difficult to conceptualize what this spectrum is, we kind of need to take this into account and kind of have this nuance when we're trying to find the cell states. This plot is actually quite interesting. What you see in the bottom is like, normally we will have an optimal state where we're going to have a specific cell stages, but in most cases, cells are compared into different perturbations. This can be infection, this can be cancer, or this can even be physiological stages like cell differentiation or cell proliferation. So it's always something to take into account. So in general, what we're trying to do here is to bring biology into our data. Until this point, most of the things that we've been analyzing is just a matrix of cells by genes and counts. The idea of this is trying to interpret all of these genes patterns into into biological information. There is a lot of challenges in here. Annotating cells on the single cell expression is quite challenging in the sense that, well, what is a cell type? So that's still a very defining question, and is even understanding the history of what a cell type is, it's important. Until maybe 10 or 20 years ago, cell types were defined by multiple approaches. They could be functional validations, they could be microscopy, they could be theoretical knowledge. So combining this with what we now consider a cell type at the molecular level based on the transcriptome, it's also a complex question. And for this, it's also important to take into account that gene expression is not decreased by a continuum, as I mentioned before, and gene expression doesn't necessarily mean cellular function. Some things that we might know by protein expression or by functional characterization of a cell doesn't necessarily going to reflect at the transcriptome level. And the other thing to take into account is that the resolution. For example, in this plot that you see here, you can see a TCE in this case of main major populations of the immune cells, where we're going to have CD4 T cell, B cell, monocytes. But we can always select a subset of this population and even subset it even more. perform our clustering, and we're gonna have then subtypes of, in this case, for example, CD4 T cells. But if we even increase the resolution, we're gonna have different functional subtypes, for example, which we can have, for example, what allergic specific Th2 cells, superpathogenic T cells. But this basically means that we kind of have to have a balance into like how much resolution we have and how much discovery we're going to have. So this is always an important, and in a lot of the cases, it comes not only to the biology, but also what biological questions do you have. If you're interested in difference at the major subtype level, you might stick into the major populations. But if you're actually interested in specific changes of a specific populations of a specific subtype, you may need to increase the resolution on the dimension, meaning that you may need to use more markers and more nuances of changes. And I guess here's also the question of like, what is actually a cell type and what is a phenotype? And it's also very arbitrary. It depends on the question and on the biological model. So it could be a phenotype can be considered a cell type, but in other cases, if it's reversible, it will just consider a phenotypic change. Okay, if a cell is inflamed, it may still come back into their own original state. At the same time, it's compared to what is a differentiation state. So if it's a developmental differentiation stage, or if it is an infection-based state differentiation, a transcription factor differentiation, these are all factors to take into account. And in a lot of cases, really depends on the biological model. Let me change. So there's different approaches to do a annotation of this cluster that we identify in the previous approaches that Orr mentioned. I would say like some of the three main approaches are based on non-marker genes, based on differential gene expressions, and annotating on previous reported module scores. So this is usually if we're doing the novel cluster annotation. If we have and defined cluster and we kind of want to know what is there, we can use these comparisons of differential expressions that are used. And defining these genes, we can try to interpret what's there. There are certain edge cases that we kind of have to also take into account, and it's, for example, doublets, which you evaluated a bit with Daniel and Daniela before. If there's low quality cells, meaning that in a lot of cases we're going to have cells of low, either apoptotic cells, which are biological low quality cells, or technical low quality cells, which will seem as a different cell type of a different cell population, but they're not necessarily are. And then host pathogen annotations are also something important to take into account. So in a lot of cases, we might be working just with one specific species, let's say human or mouse, but in other cases, we might have pathogens, which can be parasitical pathogens, viral pathogens, bacterial pathogens. In the case of viral and parasitical that have a poly-A, we can also detect their transcriptome when we do this, meaning that we also can take into account the parasite transcriptome for this annotation. And we can also identify if a cell is infected or not. And the next question, which we're not going to go that in depth here, but it's important to take into account, is the clustering accuracy, which is related to the previous question of what is the resolution. And basically, we need to have a balance into over clustering or not clustering. And in general, this is going to be an iterative process. We're going to try to evaluate, do we have too many clusters, meaning that at some point, two different clusters are basically the same, and the patterns of expression are too similar that it doesn't make sense to split them into two clusters. So what is it required to understanding the specific cell types and their markers? So biological expertise is critical. A lot of these things are biological. It's based on biological knowledge. You will end up with a list of 10, 20 genes. And a lot of the cases, they might not like be immediately obvious what they mean. So this is really requires a lot of biological knowledge. A lot of the time going back to the textbook, going and look at the internet. I would say also something I think I mentioned before, but I would like to really highlight is many protein and RNA markers overlap, but a lot of they don't. So this is because we have a lot of cross-translational and transcriptional modifications. That means that the amounts that we have at RNA doesn't necessarily reflect at the protein. And this is important because a lot of cell types are defined on antibody markers. And if you go and look these antibodies at the RNA level, they don't necessarily overlap. And in a lot of the cases, they just don't work. So it's important to be a bit critical. So if something doesn't work, that doesn't mean it's not that. So if you have a cell type that says that CD40 is an antibody that defines this population, it's always been canonical, known, and every time someone does a flow cytometry analysis or immunostaining it always you can use it to identify. Doesn't mean that when you do this single cell transcriptome or a special transcriptome analysis, you're gonna find the same marker at the RNA level. And there is some experience, but this is something that if it's a new model or a new system, you might have to figure out at that moment. It's very easy to annotate cell types and cell state. Easy is a strong word, but it's very difficult sometimes to annotate pathological and cell states. And the best way to put this into account is that a lot of these cells are just aberrant stages. They are similar to the original cell state, but they have a lot of messed up stages of signaling and differentiation, which will mean that whatever you've been expressing doesn't necessarily make sense to what we know biologically. And this, for example, happens all the time. You may have an immune cell that is expressing brain cells, brain markers, and it's like very confusing. But if you think into what's going on behind, is you're likely during a pathological state or an infectious state, messing up a lot of signaling system that will mean that a lot of things that normally not expressed in a cell are going to be expressed. But also makes our job more difficult, because that means that we will have to figure out, a lot of them just don't figure it out, but at least know which ones are not defining of a cell type state. So yeah, so in general here, I'm just going through a lot of points. We're going to go into more of detail into how actually practically do this. But in general, things to take into account is normally a lot of these cases where we're trying to evaluate one population against everything else and trying to see what makes them different, what is upregulated, downregulated, or in general, what is expressed in one place and not in another. Non-marker genes are usually highly expressed and should be in the top list of genes that are highly expressed, but doesn't always mean that. A lot of the cases, the top gene expressions may be important, but the canonical ones and the ones that are well known in literature might be the next 10 or like the next 20. So it's important to be a bit open-minded when you're looking at these genes. Normally the top 10, 20, or 30 genes are more than enough to give you an indication of what you have there. But it also depends on how specific you are going to. If you're only comparing a specific subpopulation of a T cell, you might need more genes than that. This is an iterative process, so you probably will not expect to identify everything in the first pass. You likely will have to subset, merge, and evaluate more populations as you go. And basically identify how similar one cluster is to another. A lot of these also supervised approaches are not perfect, and that also means that the boundaries between a cluster might be slightly artificial. So you will have to evaluate both biological and by doing differential expression if it makes sense to keep creating subpopulations. And basically, yeah, as you go, you will have to like evaluate in every step, does this make sense or not, both biologically and database. So how do you go about finding module scores on different gene markers? One big suggestion is go to supplementary tables of a lot of papers. Usually you will find a lot of markers or modules of different sets of marker genes that you will find there. Always make sure that your species and systems are correct. Even though a lot of things are conserved across the evolutionary line, cell type markers from a mouse don't necessarily will match the ones from a human. And if you're working with even farther model species like Drosophila or C. elegans, these cell types might not have exactly the same markers. A lot of things are conserved, but not all of them. Technology might have some influences. If you're working with 10x genomics, with SQL, or with SmartSeq, the depth and amount of genes that you can detect are different, which means that you will have some certain influence on what you are detecting. This also means in different omics. If you're working with single cell proteomics, single cell metabolomics, single cell transcriptomics, your markers will be quite different. And in general, everything that I have mentioned here, I shouldn't be using the vacuum. You always try to convince yourself of what you annotated as correctly. So you kind of have to take everything into account and also take into account the context of what you're working with. And also, like, well, the other thing that I mentioned before was host pathogen annotations. And this is important because if something has a poly-A, you will detect it, meaning that if you have a virus, a parasite, a fungi, anything that is intracellular and will have a poly-A, you will also capture that RNA, which means that when you are doing your alignment, as we discussed during the pre-processing workshop, part of the workshop, you normally will use the genome of the host some cell, for example, the human or the mouse. But if you're working with a pathogen, you also need to include the genome of this pathogen. And that will mean that whatever you detect inside of the cell will also be aligning to that pathogen genome. That said, as happening in a lot of diseases, you cells die, RNA gets released. So you will have a lot of pathogen RNA that will be detected, but that necessarily means it's an intracellular pathogen presence, which makes things very tricky because depending how clean your sample is, you might have a lot of ambient RNA, and this might make a bit difficult to define if something is infected or not. And also doesn't mean that it's an active infection. You may have process like phagocytosis or even just the environmental RNA, which might imply that there may be an active infection there, but it can just be capture of the RNA. An example that we will see is the data sets that we're working with, where we have COVID-19 samples. And in this one, we're able to detect, well, we can annotate what the PCR positive samples were based on the metadata of the samples. So what you see here in this UMAP is all the different populations that we detect from the epithelial cells, from the nose, but as well some of the immune cells. But then we can actually also annotate different other metadata, like the peak level of respiratory support that the patient had when we obtained the samples. But then also based on the transcription levels of SARS-CoV-2 that we detect per the cell, we can define if something was infected or not. And this will allow us not only to define the effect of the different parts of the virus and how this align into the sample, but try to evaluate this with the severity of the disease. We will see during the data, but we can also then, if we identify which cells are infected and which cells are not infected, try to find different expressed genes between infected and bystander cells, which can give us a lot of information about the immune response and epithelial interferon response to the infection. We will evaluate a little bit of this during the practical, but feel free to ask us any questions. So going a bit more into other type of approaches. So this, everything I mentioned before, were tips and kind of a guideline of what to do when you have a marker gene list. But in general, there's other approaches, and it's actually used in literature to the maximum extent. And in this, we have like databases and reference atlases. I will kind of try to split this in two subsets of approaches. One is just using the marker genes and databases, as I mentioned before, and going into the literature. And the other one is using automated tools. So the first one is literature. This is very important. It might seem very obvious, but I would like to emphasize it a lot, and it's that a lot of this knowledge is already known. And this comes to going back into the textbook and reading against your model a lot more, see what is known. For example, for immunology, this is a field that even before single-cell transcriptomics existed was already single-cell biology, as people were doing flow cytometry, FACS, a lot of things have been characterized this way. So always try to go into the literature, try to figure out what is known. Is there signaling available? If there's immunohistology known, all of this can be used as a starting point. And try always to rely on experts and collaborators. A lot of this knowledge is new, and that's part of the point where we're trying to do these atlases, and is to generate new knowledge, but a lot of things, there are experts and collaborators that might know a lot of this. And I think it's important to rely on them because it's difficult to be an expert in every single cell type and every single model. There's a lot of databases out there, even if they were a bulk expression of a different ledger. One of these, for example, is the Protein Cell Atlas. I have a small video here of what you could do here. For example, in here, you can look for ACE2 and entry marker for SARS-CoV-2, and you can see in which tissues are they expressed. And in addition, if you go into the gene, they also have some single cell information, and they can actually, if you go and scroll, you can actually see not only in which tissue they are, but in which cell types have been reported from their data set. Same well, there's other websites like the Immunological Genome Project, the Genotype Tissue Expression, which will have a lot of information about tissue data, and in some cases about cell type data. And this is important already to narrow things down. If you have a lot of genes that are only expressing one tissue, you can narrow down to look only for that tissue. We have other we have other databases for non-model organisms, as well as model organisms like the mouse, the fly, C. elegans, zebrafish, which you can see here. And in general, it's always perfectly acceptable to just go back to ECVI or EVI databases and try to look into papers and what the genes have been reported at. We also have single cell atlases. The previous one that I mentioned were bulk-based, but we have other data or other more specific atlases for single cell data. So we have the Human Cell Atlas, which is hosted by EVI, where you will find a catalog of most of the projects done by the Human Cell Atlas. We have the Tabula Muris, which is a multi-tissue mouse atlas or dataset done by the Chan Zuckerberg Initiative. We have the Broad Institute Single Cell Portal you see here. which is hosted by the Broad Institute, and where you will have hundreds of, I believe we're in the hundreds of data sets of single cell data, and you can explore different genes and different cell types and look what's been expressed in these in different studies. And other ones are Panglao database and cell markers, which actually designed for evaluating markers and seeing which cell type they're expressed. For example, in Panglao database, which looks like this, it's a very cool data set. It's mostly for human and mouse, but the cool thing is here is that they have multiple samples, multiple tissues, multiple cell types, and you can approach it in different ways. You can put a gene, for example. So if I put here ApoA1, a gene that usually expresses in enterocytes in the gut, it will tell me which cell type I express. As I mentioned, enterocytes. But not only that, you can go even deeper and look into the data sets and see, well, what was they express. I can try to see the UMAP original from the data set and see what was the pattern of expression for this. And then for example, this is the original UMAP from that study where those enterocytes were presented and they have high expression of that gene. But I can go a different way, and then database also has, if I go into data sets, I can also look for reported markers for a specific cell type. So if I go for example, microglia, I can see in each study what genes have been expressed as marker genes for the microglia. And these are very good information that can be used as a starting point, or even more than enough to annotate your populations. And then we have the next part, which is automated tools. And this is a set of different algorithms that have been designed over the last part of the decade that allows you to do knowledge of cell types that is already available publicly or published in different reports or data sets that you might already done as a reference, and trying to use that information to transfer or extrapolate into your own data. Basically, you can think of this as a cell type like BLAST method. If you remember BLAST, the basic like alignment tool for genes, where you can put that sequence and it will tell you what's the most similar gene. Something like this we can do. We can put our cell type from our sample and we can try to figure out what is the most similar cell type from an atlas. There are different approaches, but in general, you always need a reference cell type information. So if you want to transfer from somewhere, you kind of have to have a reference. Can be doing transspecies, so that's quite cool. You can try to transfer information from a human to a mouse, from a human to a monkey, or even farther in the model in the evolutionary line. Just keep in mind that not everything is conserved, so you will have a lot of noise the further you go. In evolutionary differences, you can also do in between different omics. Again, keeping in mind that they make things that are loose in the different layers. So if you're working with protein and RNA, you might be having differences because of post-translational modification. Same thing if you do a tag an RNA, there may be difference between accessibility, gene expression. And then there's multiple different ways to do this. There's correlation-based approaches where you try to correlate the patterns of your clusters against the reference, and then supervised classification where you use machine learning in most cases to do this. This is a list of some of the tools that are available. This slide will be available for you, but in general, the could be split between the correlation based and the supervised based. They're also available in multiple languages like R, Python, MATLAB, and in a lot of the cases they have their own web app when you can just upload either your matrix or the modules of genes differentially expressed, and it will try to give the most similar cell types. So in general, yeah, so you will have like a query, which will be your data set, and you will compare these as, I guess, a reference. One of those tools, for example, SCMap, as I said, is a correlation base. So you basically will see the correlation of your cluster here and see which one of the cell types in the reference correlates the most. So in general, I will say like an overview of how would you do this and how we'll normally approach it. I will normally start with automatic annotation, either using one of the correlation of supervised approaches, just to have a general idea of what I have in my data. A lot of these methods are not perfect, but in general, it gives you a global idea of what you have. Then what you will try to do is so because this is independent of the clustering a lot of the time, the boundaries between a cell type and the other are not great. So I will then use manual annotation using either my marker genes, my gene function, and my clustering to actually define this, to try to find what are the actual boundaries between my sample. And then this is actually an iterative process. You can always go back and do some automatic annotation, and you can also get some marker genes from what is published in these data sets. And then what you will do once you have your cell types is actually you go to the verification step. And this is important to take into account, is that if you are reporting a new cell type, that hasn't been reported before, you likely will have to validate it. And this implies different experiments for functional validation, have enough statistical support to support the claim that you have a new cell type, and consulting with experts that what you have makes sense. I'm gonna go with a quick example with with one of my old data sets, just for a general information of what we are gonna see here. These are colon and ileum biopsies where we grow organoids from, we infect them with the SARS-CoV-2, and then we analyze these organoids with single-cell RNA-seq. Basically, what we had was UMAP like this, where we have around 18 clusters. And the first thing we tried to do was using this paper that was already published from the Shelek and Rejek lab, where we have around 300,000 cells from human colon, trying to use as a reference starting point and use an automated annotation. And what we have is like using our unsupervised clustering and trying to find the annotation from the published data set. We can see, for example, from this paper, the enterocytes in which cluster of my unsupervised they map. And same with every single cell type. And this can give me a starting point and an idea of what's the population I may have. But as you can see, a lot of my clusters, for example, cluster 0 gets split between multiple populations. And same thing can happen, like some clusters are basically only mapping to one. But because of these boundary differences, I will then go back into my cluster and use this as a reference. The other thing that I can do is using correlation base, I can compare whatever I found as my cluster cell type with whatever was present in the tissue atlas and try to see what's the highest correlation. And what I can see here is, for example, my enterocyte cells correlate the most with my enterocyte cells in the other one. And same so I can do with other cell types. And then the next thing I can do is then actually go back into marker genes. So for example, I can see here BES4, which is a marker gene for BES4 enterocyte. I can see FAP1, which is a marker for enterocyte. I can see markers for stem cells. And in general, I can also go back into the map and evaluate if this makes sense. For example, THGB is a marker for enteroendocrine cells, and it's only present in this cluster. That if I go back into what I predicted, my enteroendocrine cells will be mapping into that specific cluster, which was cluster 17, that you could see here. So then with that way, I can start to validate my automated annotation makes sense and in which cluster I will do. So it might be that my automatic annotation will say that enteroendocrine cells might be here, and may have 1 or 2 cells here. But because of my supervised clustering, I can close that boundary and define it just as this population. Same thing I can do with other markers like CLCA2, which is the marker for stem cells. And I can also try to do comparative markers like LGR5 and SMOC2, which are markers for stem cell. I can see where they overlap, try to find different ways where my cell type might be present. And in general, what I can do is once I have that, and I want to have my annotation, I can even do like differential expression between one cluster and another. And normally you will kind of have this diagonal in your heat map. And then if you remember at the beginning of my part of the presentation, I mentioned that you can have low quality cells that might be blurring your annotation. In this case, for example, I have 2 populations that I call low quality enterocytes, that if I go back into my data, I can see that they have low amount of transcripts, a low amount of counts, and they're likely a technical and a mix of technical issues, but also a biologically apoptotic population of cells that actually have because they're lowly detected, will have some will be clustering on their own, and they will not be able to be annotated as a specific biological cell type. So that's basically the example that I have. If you have any questions, please feel free to ask. Happy to reply.
Thanks, Sergio. Any questions for Sergio? Okay, I think we're due to have a break actually right now. So 15-minute break. We'll resume with Orr and Sergio to go over the R lab subsets, cell subsets. I believe that is the hands-on part. So we'll go into breakout session rooms. Is that correct? Perfect.
Yeah, that's right. Christine.
Alrighty, so why don't we take 15 minutes and resume at 8:30 Eastern, 1:30 GMT. See you soon. Okay, everyone. Welcome back. We will now go into breakout session rooms. So, or I'll open up 2 rooms. Is that right? Or do you want to just keep everyone here?
I count only 8 students, so I think we can just keep everybody here actually.
Okay, so we'll keep everyone here, so no need to leave. So, or should I pass it on to you for
Sure.
the next one? Okay,
Yeah,
great.
absolutely.
Thanks, Or.
Okay. Yeah, sure. So hi everyone. So now we're gonna do hopefully in our sort of session where we will get to practice some of the concepts that just got introduced in the last hour. So go open your our studio server. I know you've done this a few times already for Daniela's lesson and Sarah's and Daniel's lessons. So you're going to open up our studio server, and you're going to open up the file called HCA COVID-19 Clustering Lab. And so what we're going to do in this lab is take some of the data from the Shalik lab, where they had a study of COVID-19 infection. And we are going to just, load up that data, do clustering on it, and see, what are the cell types and subsets that we can identify. And, if we're interested, we might do some more focus questions about that. So hopefully you can all open up this script. Because we don't have many people here today, we're not going to divide you up into groups to do different things. I think what would probably make the most sense is we just focus on maybe just one of these activities, like interpreting PCA. So don't worry about this group stuff for today. If we had more people, we could split up, but I don't think with this few people we should split up. So what the script is going to do is it's going to take your data in from this COVID-19 study, and we're going to carry out identification of variable genes, principal components analysis, clustering, visualization using UMAP, and just kind of making some informative plots about how the data looks once we've done all those steps. So everything should be able to be run just as is. To remind you, what you're going to want to, maybe one of the more important things to do is you want to make sure that your directory is set up. So if you go to line 29 of the script, there's this little piece of code that says where the results are going to be written to. And it's going to be your home directory, which I think you've seen this before, but you can click on the bottom here and go to the terminal, and it should say, you know, for instance, my computer is SCW90, but if you're not SCW90, you're probably like SCW01 or 5 or whatever. Just replace this line or replace this part with whatever user ID you have. So then once you've done that, every chunk of the code can be run just using these green arrows that say run current chunk. And all the code is written out already. So the focus of this code is more complicated than what you saw before, or you've seen in the previous labs. So really, we want to focus more on what the code does and what are the plots that are made and how to interpret them. More on that and less on like necessarily understanding all the syntax for this code and things like that. One very helpful thing I'd like to remind you all about is that, you know, there's going to be a lot of functions you'll see, like create Seurat object or normalize data or find variable features. So these are all different functions, and you can learn about them just by writing question mark in the name of the function. So here you do create Seurat object with a question mark at the beginning, and there should be some documentation that will appear on the right. So create a Seurat object. Let's click on that one. And eventually will load, and you can see here like description of the function and the arguments, and then what each argument actually is. So that's always a good first step before, you know, getting frustrated, is to just look at the documentation. So what we're going to do is there's these different code chunks that, you know, they're very described what's in them. So all the main code chunks have the word main in front of them. So main feature selection, main PCA, main clustering, main DE genes, differential express genes, and then that's it. So what you can do is read through the lines of code and the description. And as you read it through, you can run every single chunk by just pressing the green arrow. And we'll do that. And I'll give you all like maybe 15 or 20 minutes to start working through that. And you know, what you're going to want to do is run those lines of code and try to understand what the outputs are at the bottom. And again, if you have questions, you ask me or Sergio or Adam, who's nicely joined us today. Or you can put this like, you know, question mark type thing into the console and look there. So, and we tried to write out kind of the logic of all these steps. So please kind of read through these things as slowly as you want. You don't have to rush through this. Okay, so we'll give you all like 10 or 15 minutes to get going. And if you have any questions, just, you know, you can just say them or put them in the chat. Me, Sergio, Adam, I think Vincent and Collins are around, and Dominic. So yeah, we have plenty of people who know a lot. So please ask us as you go through things. And are there any questions overall, any general questions overall, or things we can clarify before you go? Get going. Great. So we'll check in with you all in 10 minutes, but please ask anything before then. We're here to help. Okay, I think I'll just check in with you all and see how things are going. Does anybody have questions as they're working through this?
I do.
So, yeah.
So I was actually wondering regarding the PCA, you have said on your on the class that you normally do like a lot of them, like 20 or 50 or something like that. But it seems that at least on mine, it has just run 5 times. Is that correct?
So you're saying in the code here, it ran 5 times?
Yes.
Okay,
Like
so good question,
PCA
Isabella. So
1. Mhm.
how about I walk everybody through to that point and then we discuss that point. Okay,
Sure.
thanks. I'll get right to it. Okay, so I think it's worthwhile to just talk through the code since I know maybe all of you or most of you have not coded before, so it's always good to hear this multiple times. So the first chunk of code, like I said, we can just run with this little green arrow. And the first part is just loading up the different libraries we need. The main one is Seurat. Then we set where the results are going to be stored to. In my case, it's this directory called SCW single cell workshop 90. And then the main part of this code in this chunk is reading in the data. So we have both the counts data, which we read in from this thing called an RDS file, and we use the read RDS function. And this creates a matrix called counts. And it's a really big matrix. It's 32,000 rows and 6,000 columns, which are the cells, and the genes are the rows. So we're reading the counts, and then we also read in this metadata, which you can look at here. And there's different for every cell. There's different pieces of information, or what we call metadata, about that cell. So this sort of metrics from the WHO, percent mitochondrial, the PCR status, and so forth. And then finally, once we have the counts and the metadata, we can use the create Seurat object function to create a Seurat object, and we see the description of that in the documentation. Okay, and then you have the Seurat object called GC data. And you can see it's an object of class Seurat. There's 33,000 features, or what we call genes, and 6,000 cells. Okay, and then finally, we add this little extra thing to the metadata, which is going to just give us the status as to whether this is a control patient or a COVID-19 patient. And that's just doing some string parsing, which you don't need to worry right now about how that code works, but happy to break it down if anybody's really interested. Okay, so that's just some housekeeping to load the Seurat object. And then the next section, the next chunk, it's called selecting highly variable features. What we're going to do is normalize the data, which, as you remember, means that now every cell has counts that sum to 10,000. That's this, and then we take the log of those values. Then we do find variable features, and we look at what those features are. And features and genes are the same for the purposes of what we're doing right now. So I just hit run on that little chunk. The data gets normalized. You find variable genes, and yeah, and then you get these nice little plots. And so this is pretty similar to what I had shown you in the lecture, where you have a plot here where it's the average expression and some measure of variance. And as I said before, there's an inherent relationship between the mean of the gene's expression and the variance of the expression, because they're following a distribution that's sort of like a Poisson, but not quite. So the higher the expression, the higher the variance. So you need to kind of correct for that. And these genes in red are the outliers from the bulk. So there's 30, I mean, it's hard to see this because it's over plotted, but the lion's share of the density of genes is here in black. And then there's 2,000 genes that are kind of outliers, and some of them are labeled here. Okay, so that's how you find the variable genes. And this is just some output there. Any questions on that? Okay, then let's go to Isabella's area where she was asking questions. So now that we have the variable genes, and those are stored in this in this variable, you can look at them here. This has all the variable genes. If you just do variable features, and we can see how many there are. I mean, there should be 2,000 because that's, I think, how many we requested. But there's 2,000 variable genes. So now that we have the variable genes, we've gone from 32,000 dimensions, which is the number of sort of features or genes in the data, to, and it's more than 20,000 because there are long non-coding RNAs and other little things in the reference, but we've gone to 2,000, and now we're going to further reduce the dimensionality using PCA. And how we do this is first, we're going to center the data around 0. And this is a mathematical transformation that we commonly do before doing PCA because it makes the it makes things interpretable. So we're going to center the data around 0, which means every gene's expression before it ranged from 0 to some high number. Now what we're going to do is we're going to take every gene and subtract off the average expression of that gene across all cells. So now every gene's expression will be centered around 0. And this is, like I said, a transformation that you do because it makes the principal components analysis much more interpretable. Okay, so we scale the data, and then we're going to run the PCA. So we just run that little chunk here, scale data, and run PCA. And so now if we look at, we have several ways the data is stored right now. We have counts, which is like our raw data. This is the UMIs. So you see these are like 1, 2, 6. These are like the UMIs, the unique molecular identifiers. Those are always integer number counts. And now we have our normalized data where we've summed things and done log transform. And so now these are non-integer numbers. And then we finally have the scale data. And when we scale things, you see now every value, every gene has a value across every single cell. And we've subtracted off the mean. So every row here is a gene and it's a measurement across the cells. So if we were to take, like, say, the mean here. Actually, I don't think I need to use it's already, no, it's not sparse anymore. If we were to take the mean of every row, we'll see that it's like very close to 0. So all these values is the mean expression for every gene now. You can see it's very close to 0, like 10 to the -3, 10 to the -17, and so forth. So that's what scale data does. It centers every gene's expression around 0. And then we've done the run PCA. And the output of the run PCA is kind of below here where we have PC1, PC2, 3, 4, 5. And so Isabella's question was, why are there only 5 PCs? It's a good question. So this is like more just output that gets sent to the console when you're running run PCA, but it's just part of the, it's just a subset of the PCs it's showing. It's actually way more PCs are stored. So if we go to the run PCA documentation, we can see that the default number of PCs that it calculates is 50. So if we go to NPCs down here, total number of PCs to compute and store, 50 by default. So this is just showing us the first 5 PCs and the genes that have very high loading and very low loading on those. So these are like, you can think of these as like programs or genes that tend to be on or off together. So, you know, this gene to TUB-A1A and RRAD and SES1 and OMG tend to be high together. And these genes, EMP1, SCEL, tend to be low together. And similarly, if you go to PC5, SciBB, and TyroBP, and TGF beta I, and so forth, these tend to be high together. And these other genes tend to be low together. So there are more than 5 PCs. We can see this if we access the PCA slot here. So this is just a way to access the PCA dimensional reduction calculation. And we can see the embeddings of the cells, which let's just take the first 5 cells. And so here are the first 5 cells, Isabel. And so this is a cell, and then this are the PCs that were calculated. PC 1, 2, 3, 4, 5, 6, all the way up to 50. And so if we had set the number of PCs to a different number, we would have had a different number here. But it is actually 50 PCs that got calculated. And we can see the overall dimension of the data of the embedding of this matrix. So now it's 6,147 by 50 versus the matrix you started with, the scale data, which was the input to the PC, is 2,000 by 6,147. So we've gone from 2,000 genes to 50 principal components. And the principal components are defined as groupings of genes. They're linear combinations mathematically, where different genes have different weights. And these are the most positive weights, and these are the most negative weights for PC1. But in PC2, the weights for all the genes will be different. And remember, the PCs are capturing the variance in your data. So Isabel, any other questions about the PCA, or does that make sense what's going on?
No, it
Isabella?
did. Thanks.
It makes sense. Okay, thank you. Great, good question. Yeah, this is just, again, it's just like what Surat's kind of throws out there. Okay. Do people have other questions? I can keep walking through this or give people time. If anybody has a strong preference, let me know. But any questions? Maybe people are still here, right? Let me just see. Yeah, people are still in this thing. Okay. Okay. I can just keep walking through the last 2 main chunks of this, and from there we can talk more about questions. But okay, so now we've done the PCA, excuse me. And remember, now the next thing is we're going to do clustering, which involves creating this pairwise distance matrix between every pair of cells. So what that means is, we'll take this cell and this cell here, and we'll calculate its distance in the PC space. So we have 50 measurements for this cell and 50 measurements for this cell, and we just get the Euclidean distance between them. And we do that for every pair of cells. And if 2 cells are very similar to one another, they'll be close together in PC space. And if 2 cells are very different, they'll be far apart in PC space. So this will give us a measure of similarity of these cells. And now we're doing this in a much lower dimension of 50 dimensions. Okay, so we'll start with these first 2 lines here. I'll just enter them in one at a time. So first we're going to compute the nearest neighbors graph and then the shared nearest neighbors graph. So what this function does is we can look it up like everything. Find neighbors. So it computes the nearest neighbors for a given data set. So what this does, right, is we're going to make the distance matrix. And then from the distance matrix, we're going to get the 20 nearest neighbors. That's this K. So it's often called K nearest neighbors. And then we're also going to compute the shared nearest neighbors graph. That's SNN. Okay. So now there's also a graph that's stored in your Seurat object. Let me see, I don't actually know how to, it might be in your graphs, let's see. Yeah, okay, so let me just get. Okay, so there's two graphs now that are stored in your Seurat object. One's called nearest neighbors and one's called shared nearest neighbor. And you can kind of look into them if you're really curious. I haven't looked at this myself, but these are basically let me just actually make this much smaller. So what this means is that there's this is like a representation of a graph where you have the cells as your rows and columns, and then the values are whether there's an edge between the two cells. So here there's an edge between, this cell and this cell. Okay, any questions on making the graph? Okay. And then the next thing we'll do is do the clustering once you have the graph. So that's going to happen on the shared nearest neighbor graph. And we'll run find clusters, and you can see there's actually at least two algorithms you can choose from and find clusters. So if we go to find clusters, there's this algorithm argument or parameter we can set. And this is the algorithm for modularity optimization. So a few years ago, most people were using Louvain, and now more people are using this algorithm called Leiden, which has some mathematical features that make it better. But it's not a huge difference either. So by default, I think it uses Louvain. So you can see the defaults by just scrolling up. It's algorithm equals 1, and that algorithm is the original Louvain algorithm. So we do the clustering, and then we'll make a UMAP that shows the different that will visualize all the data, all the cells in 2 dimensions. So that will run. Oh, it's done. Okay. And now we have our 2 things we need. We have a visualization in 2 dimensions. This is the UMAP. And then we also have the points labeled as clusters. And the clustering is stored in the metadata now. So if we look at the metadata, which we can just access with this bracket kind of syntax, we see that now there's all this metadata still, but there's a new column at the end called Seurat clusters. And every point is now assigned a cluster. And we could access that also by just typing Seurat clusters here. And yeah, and so we see, different cells have different cluster IDs. Okay, so now we can plot that using this dim plot function. So this will plot the UMAP, and it will label the clusters with whatever current identities we have loaded in. So we can see that by just going to plots, and now we have this UMAP embedding where all the cells are in 2 dimensions. Each point is a cell, and things are now colored by the cluster. So 0, 1, 2, up to 16. Okay. So we can see certain clusters are pretty distinct, like cluster 13 or cluster 4 or cluster 14. And then other clusters look like they might be part of related cell types or states, or maybe it's a continuum. Like these look like a pretty big group here, like 0, 2, 3, 5, and 6, and 12. Similarly, like 7 and 8, they kind of look like they might be a group, and 1, 9, 11, and 16. And so I'll just repeat that the UMAP itself is its own technique that's trying to map these cells in high dimension to a point in 2D space where similar cells are near each other and dissimilar cells are further apart. But the clustering is happening with the series of steps we had discussed. So it's PCA, building a distance matrix, building a nearest neighbor graph, and doing clustering on that graph. So there are two independent operations, building the UMAP or the TSNE, which is really for visualization, and the clustering, which is really this sort of very quantitative way to say which cells are probably in the same cell subset. So they're doing sort of similar things, but it's not like the clustering is not happening on the UMAP. The clustering is happening using the PCs and building these graphs and so forth. However, the clusters will often look like they highlight specific regions of the UMAP, and that actually makes sense because the UMAP is trying to preserve distances from the high dimensional space into the low dimensional space. So yeah, so this is something that's a little subtle, but I just want to bring it up. Any questions so far on how we got the UMAP and how we got the clusters? And don't be shy. If you're totally confused, that's fine. We can take a step back.
No, I just have another quick question.
Sure.
When I run it, I did got my UMAP, but on the code had an error in grid new page. Is this normal?
Where did you have an error, Isabella? Which line?
After it run the UMAP,
Okay.
and then the UMAP was created, and
Yes.
then after the GG save,
Oh, it didn't like
paste,
that.
blah blah blah. Yes, and then it got like error, couldn't open the file.
It gave you an error.
I can copy. Yeah, I can copy
Yeah,
here.
put it in the chat. Just copy the error and put it in the chat. So this is probably, it's not going to be a very interesting error. So sometimes there's issues with the what's called like a graphical device, which is what's rendering your plots into something that can be saved as an image. And the graphical device is somewhat tied to your underlying system. Let's see. Could not open file. Okay, so I think your error, oh, this is an easy one though. So your error, Isabella, is you're trying to write, are you user SCW01? Is that your ID?
Yes.
Oh, it is?
Yes.
Interesting. Okay. Cannot open, huh. It sounds to me like something with reading and writing. I don't know why then, because that looks more of a, like not able to access a certain thing type error rather than an error with a graphics device, but it could be an error with a graphics device. Is anybody else getting that error when they try to save? Like when they run that GG, when people run this line, do they get an error? Nobody else. Did anybody not get an error? Mine is working fine. Yeah, you can share your screen. Isabella, do you mind sharing your screen for like maybe 10 seconds? I can just.
No, it's fine. I just need to open here.
Sure. It works for Daniel. Okay, I think we can.
Is it
probably...
working?
It works for some other people. Let's just see. So if you can, you just for my sanity, can you click on terminal for a second, just to the right of console? And your SCW01 for sure. Let's just. Okay, you're SCW 127 actually. So what you want to do, Isabella, is if you scroll to line 20, whatever. Keep going up. It's like the first, keep going. Okay, right there. So where it says SCW01, you want to make it SCW127. And then just copy okay, yeah, you can, okay. So you'll want to rerun the chunks up until like rerun every single chunk, and the problem should be fine. So
All right.
oh yeah, Adam's point. Yeah, Adam is entirely right. And I think if we ever, well, when we do this in the future, we're gonna do exactly what he says, because this has caused like 10 problems, more than 10. But do you get the reason why this is happening, Isabella?
Yes, I got it. Mhm.
Yeah, it's basically you're trying to write to a directory that you don't have permission to. So
Mhm.
Vincent and Collins and Dominic set up everything so that every student has their own directory, and we can't write into each other's directories. Yeah. So if you don't
Okay,
mind
oh, good.
stopping to share your screen, I'll go back and
Sure.
come back to you. Great, good. Glad we
Thanks.
figured that out. Sure. Okay. So yeah, and then once this is GG save, which will save the plot so you can look at it later. And if you go to the files tab, like we were looking at a plot, if you go to the files tab, you'll see that this file is now saved, and you can click on it and it will open in your web browser. You can also download it to your personal computer, but there it is. Okay. So that's a nice thing. It's good to save our plots. It's one of the nice things about the R kind of markdown too, is that like the plots, you can integrate them into this document, or you can just save them in a folder and put them in your lab notebook or computational lab notebook or presentation slides. Okay, so next we'll just do the same thing, but we're going to take that UMAP and now color it by the course cell annotations that were provided in the publication, this COVID-19 publication. We have a file that's saved now of that as well, so we can open it, UMAP annotations. It's a little crowded, but we can kind of see what we saw before, but now it's labeled with the cell subsets that were identified, the cell types that were identified by the authors. So here's this population that's really far off called ionocytes, and then this population here is squamous cells, and then this group here where I said it looks like there's multiple cell states, or it's one cell type. This is called ciliated cells. And then this group over here is called T cells, and so forth. And so depending on what part of biology you're coming from, maybe you're familiar with the lung and the airways, or maybe you're not, or maybe you're familiar with the immune system, and so T cells and B cells ring a bell. But no worries if these all sound like very foreign to you. These are just different cell types that are, important in the COVID-19 infection process. Great. And so what we're seeing, largely speaking, right, is, we can use the transcriptomic information to identify cell types and cell states. And we have like thousands of cells here. Like, it's actually way more cells in the paper, in the publication. We've just subsetted this data a lot so that it runs quickly for you in Seurat. And so we're not waiting for things to run. But there's a lot of cells. I think it's more like 100 or 200,000 cells in the actual publication. But yeah, what we can do, so and how these cell types were identified are using methods that Sergio had discussed with you in the last hour. Yeah, so this is great. We're seeing all the cell types that you were supposed to see when looking at this tissue type. And then we can also do some other things, like we can label the plot really by any metadata we want. So, we have all these things in the metadata. You can look at all the things that are measured in the metadata. So these are all the different things that are stored in the metadata, and we can color the plot by whatever. Like here, we're going to group it by COVID-19 status. So what you see here are the same UMAP, and now just colored by whether the cells came from a control patient or a COVID-19 patient. And you see that a lot of these clusters have cells from both. both groups, and some clusters seem to be predominantly one group, like this seems to be mostly the control patients, but yeah. And then what was that cluster actually? Let me just go back to the, so that cluster is which is that the squamous cells? Yeah, I'm not actually sure why it's just yeah, actually maybe Sergio knows or somebody why it's just squamous cells in the control. But either way, that's what we're seeing from the data. Like most of these clusters are having cells from both the control and the COVID-19. And then, like what you would want to do biologically speaking, right, is take these clusters, like say, take the T cells and compare T cells from the COVID-19 patients to T cells from the healthy controls and ask, what are the transcriptional differences between those, within that cell subset when you have this SARS-CoV-2 viral infection. Any questions? Things we can clarify about that? Good. Okay. Let's keep going then. And then the last thing I kind of visualize is the amount of detected SARS-CoV-2 RNA. Oh, is a question? Okay. Okay. Great. Thank you. Thanks, Modena. So the last thing we'll visualize is just the amount of SARS-CoV-2 RNA on a per cell basis. So this part is just to convert the values to integers because I think they start as strings, which isn't good for plotting numeric values. Yeah, so they're all strings here. So I just converted it to an integer to start. Now you can see that everything is integers. And then most are 0 because we don't do a great job of detecting viral RNA, and also not all the cells have virus in them. But now, regardless of that, let's just plot it. Okay, and so what we can see is this grouping of ciliated cells down here. We detect SARS-CoV-2 RNA, and then also in some of the cells here. But for most of the cells, we're not like really picking up the virus. But that's not surprising because not all the cells get infected, and this is also a hard thing to detect, just in an experimental from an experimental viewpoint. Okay, great. So, I encourage you to like play around with that. Like this dim plot function and this feature plot function are very helpful. Dim plot you use for plotting like categorical data. So like cluster 1 and 2 and 3, or cell type like ciliated cells and T cells, versus feature plot plots sort of numerical data over your UMAP or whatever dimensional reduction you're using. So we could also have just plotted something simpler like the number of genes per cell. So this plot is just overlaying the cells, coloring them by the number of genes that were found. Actually, sorry, number of genes that were detected per cell. So some cells have higher number of genes and some have lower. Okay, so feature plot and dim plot are your friends. They are very helpful. Cool. Okay, so the last section of the script is this differential gene expression analysis. Any questions before we go to that? Okay. Feel free to put questions in the chat. Anything is good. If you're getting errors or you just completely want to discuss something earlier, that's fine. No rush. Okay. So the last section is we're going to look at genes that are differentially expressed in one cell subset relative to all other cell subsets. So what that means is we're looking for genes that are really different in expression in a cell subset relative to all others. So that could be either the gene is much more highly expressed in the subset relative to all other subsets, or the gene is much more lowly expressed in the subset relative to all other subsets. Generally, it's easier to think about genes being more highly expressed in one cell subset relative to other subsets, mostly because it's often hard to interpret zeros in single cell transcriptomic data without an additional layer of thinking. So if a gene is 0 in a cell subset, it might be because it's really off in that subset and maybe it's on in all the others. So that's informative, but it could also be 0 because of just technical reasons of just it's difficult to capture that gene and that cell type for whatever reason. But maybe the gene is actually there. Versus when a gene is highly expressed, it's still affected by technical noise, but we know that biologically it's probably on and being expressed. So that's a little easier to interpret when genes are more highly expressed in one cell subset relative to another. So that's generally the types of genes we start with looking at, are the ones that are high in one subset and low in all the others. So like I said before, there's many tests that you can run in Seurat, and we're not limited to Seurat. We could do things outside of Seurat, but for this workshop, we're just doing things in Seurat. So here are a few of the tests that are in Seurat. There's Wilcox, which we had mentioned earlier. It's a non-parametric test. The t-test relies on, data following the student t distribution and whatnot. MAST and these others are more like other probabilistic models that one can use. But Wilcox tends to actually do fine. And that's my default. It's just fast and simple to interpret. Okay, so we'll choose to do the Wilcox test. And then we're going to find differentially expressed genes for each cell subset. The identity of the cell subsets is stored in the Seurat clusters column of the metadata. So remember, we can look at the metadata. And so here's the Seurat clusters column. And what we'd like to do is say, for all cells in cluster 5, what genes are higher in cluster 5 relative to all others? And to do that, we're first going to focus on the Seurat clusters. So we're going to say the identities we want to look at are Seurat clusters. And then within each of those identity groups, so Seurat cluster 1, 2, 3, 4, 5, what are genes that are differentially expressed in that cluster relative to all others? And we use this find all markers function. And you can read about how find all markers works, in the documentation, find all markers. And these are different parameters we send that just tell the function how to run and filter a little bit of the data. Not super critical. The main thing is we're going to tell it what test to run, which is the Wilcox test. So if we look at the tests that are available, you can see there's Wilcox and ROC and the t-test and so forth. Okay, so there's a lot of things that you could do. So we'll find all markers with the Wilcox test, and then we just run that. And so you'll see for every single cluster, it's going to calculate the code will calculate differentially expressed genes. So now it's working on cluster 1 and cluster 2 and 3 and so forth. And so if you were really, had to do this for millions or thousands of single cell RNA-seq data sets, you could probably parallelize this to speed it up. But, for one data set, this isn't too bad. Okay. And then once those are calculated, and I think there was 16 or 17 clusters, depending on how you count. Once that's calculated, we're going to have a data frame called gcdata.markers, and that will have all the results from the differential expression analysis, and it will be broken down by cluster. And then at that point, you can filter this matrix, or sorry, this data frame any way you want. And so there's going to be a lot of differentially expressed genes, and often we just decide to look at a subset of them, like maybe the most highly significant or the ones with the biggest log fold change, and that's just easier to start with. Question: how do you select the log FC? Okay, so Isabella's asking about this threshold here. So this is the log fold change threshold, and this is basically saying that the we're only going to look at genes whose log fold change is at least 0.5 between the cell subset we care about and all other cell subsets. So this is log scaled. So it's really, I forget if it's log base 2 or log, I think it might be log base 2. Let's see. I'm not sure. But basically, the bigger this is, if it's positive, it means we're only looking at things that are upregulated. So sorry. So if it's the bigger the number, the more we're asking for genes that are very highly expressed in the subset relative to all others. So the biggest fold change. So the bigger the fold change, the more like probable that gene is something specific or special about that cell subset, versus genes that have very small fold change. It becomes a lot more questionable what they're doing and whether they are really that important for the cell subset in particular. So how do you set the threshold? It's very much a heuristic, meaning there's not like a number that we always set it to. We often try different values depending on how many differentially expressed genes we want to look at. I think it's easier to set it to a high value to start and then lower it if you don't feel like you're getting enough differentially expressed genes to get anything meaningful. Yeah, so there's not really a right or wrong because it's just a statistic. The p-value that comes out is important. So let's look at this GC data markers. So here we have every gene and it's ranked by the p-value, how significant it is. And here we're just looking at, say, cluster 0. So in cluster 0, these are the most significant genes, like this one, DNA H11, has a p-value of whatever, some ridiculously small number, probably a little overinflated in some way, but the fold change is 2.5, log fold change is 2.5. There's an adjustment that's made to the p-value to allow for multiple hypothesis correction. So you can think that every single gene that you're testing, whether it's different between cluster 0 and all other clusters, that's a hypothesis, and you're trying to, reject the null hypothesis that the distributions or the values are similar between cluster 0 and all others. But we're doing this for thousands of genes. Like in theory, you could do this for 20,000 genes. And so when you have multiple hypotheses, you need to correct for the effect that the more hypotheses you test, the more likely it is that one of them gets rejected just based on pure randomness. And it's kind of like the analogy that I find very helpful is, I always like to go back with probability to the coin flipping example. So imagine you have a coin that can be heads or tail. And the experiment you're going to do is you're going to flip the coin 10 times and ask what proportion of the times did the coin land on heads. So you flip a coin 10 times. The most probable outcome, if it's a fair coin, is that it lands on heads, okay? And so you would never, if you do that, you end up not rejecting the null hypothesis that, it's a fair coin. Like, let's say our null hypothesis is that the coin is 50-50 heads tails. So you flip a coin 10 times, odds are it comes out heads 5 times. That's the most probable outcome. However, in the real world, it might come out heads 4 times or 6 times. So now if we repeat this experiment like 100 times or 1,000 times, it's still always a fair coin, but 1 time or 2 times, you're going to get something weird where you only get like 1 head and 9 tails. And for that one, where you have those 10 tosses, that's one experiment, you're going to reject the null hypothesis that, that it's a fair coin, that it's 50-50 because you only got one head. So what statisticians and probability people have done is, they recognize this and, they model this with multiple hypothesis correction. And so what that does is really takes into account the number of hypotheses you're testing. In the case of the coins, it might be that you do that coin flipping experiment 100 times or 1,000 times. In the case of the genes, it's you're doing this gene expression comparison, 20,000 times. So just by chance, even things that are identical can look different based on sampling, and things like that. So that's what this p-value adjusted is. It's doing the multiple hypothesis correction, and so then the p-value becomes less significant. But these are still below, the typical 0.05 threshold. Okay, any questions? All right, so moving on then, what we're going to do is use this code from dplyr, which is a really nice package for working and manipulating data frames. It has some really nice sort of functions built in that allow you to split data frames, apply functions, combine them back, but just do basic filtering as well. So we're going to filter the data frame to only look at genes whose expression is higher in the given cluster relative to all others. We're going to group the results by cluster. So for every cluster, we're going to get a result, and we're going to look at the top 10 genes in terms of their average log fold change. So right now, the dimension of gcdata.markers is there's 17,000 rows and 7 columns. So we'll do top 10 genes. And you'll see that now there are 170 rows. So there's 17 clusters, and we're asking for the 10 genes that are most highly expressed in those clusters relative to all the other clusters. So now we can look at what that looks like. Examine that a little. Okay, so we have genes. So this is cluster 0. So DNAH11 and SYN1, and these are all the log fold changes. And these are the top 10 by log fold change. Although I don't know why the ordering is like that. But yeah, and then we could look at, clusters 11 through 20, rows 11 through 20. And that's cluster 1, and then we could look at 161 through 170. That's the last cluster. This is cluster 16. So these are all the genes that mark those clusters. Any questions so far? I know this code is maybe a little weird looking, but I really encourage you to, take on your own time. This RStudio server, I think, is kept open for another 2 weeks. Vincent will correct me if I'm misstating that, but this will be open for a while still. So you have a lot of time to just run through this at your own speed, put these lines of code into the console and see what happens. And, we'll still be available over Slack to ask questions. So don't feel like too overwhelmed right now. You have still a lot of time to look at this and ask us questions later on. Okay, so then we've got the top 10 genes. Now we're going to get the individual genes and just store them in this little character vector. This is 170 genes. We'll make sure that the data is scaled because that helps with visualization. And now we'll do a heat map, which is what we showed in the slides. So in the lecture slides, so you'll recognize this. We're going to plot the genes across all the cells, grouped by their cell subsets, and we'll have a little heat map show up. And it's going to plot. It takes a little bit of time because it's lots of cells and lots of genes. Oh, it's done. Where we go? Okay. Oh, I like these colors. Okay, good. These are a little easier on the eyes. Great. So this is gonna show up in a second. It's a little messy, so let me try to save this and maybe I can blow it up so it's easier to see. So we'll save it. And when you save, actually, what's nice is you can change the width and the height. So this can be really nice for figures or just making sure things are readable to the reader. Okay, so let's go into files now, and we'll look for this file DE genes heat map. Okay. Hopefully this doesn't crash my computer. Okay, this is looking better. Load, 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 load. Here we go. Okay, great. Okay, so from the sort of 30,000 foot view, you see on the columns are the cell subsets 0 through 16, so 17 subsets of cells. And the rows are the differentially expressed genes, 10 for every single cell subset. And you can see this sort of diagonal structure where, the genes for subset 0 are high in subset 0, and red is highly expressed, and blue is lowly expressed, and then this sort of off-white is close to 0. So you see this sort of diagonal structure where, it's high in cluster 0, but low in cluster 1. Now let's zoom in. So here we have cluster 0, and these are the 10 genes that we had looked at before. And then here's cluster 1, and these are another 10 genes. And you can kind of look through the genes, and, Sergio talked about how there are sort of manual annotation approaches and automated annotation approaches. And, the manual ones involve often looking at these individual genes or gene signatures. Interestingly, we have a cluster that seems to be defined by the SARS-CoV-2 viral genome being expressed. So I think that's in those ciliated cells that we had seen earlier. The small number there, these are probably those small number of ciliated cells, and you can see those viral genes are really low in all the other cell subsets. They're basically not detected. But that's cool. I'm not really an expert in the airway. Obviously, not obviously. I mean, these are obvious ones for me, and that these are immune genes, CD74, IL-1 beta. Some of these here are, yeah. So, I mean, you can kind of stare at these, and it's fun to look at, especially if it's your own data that you spent months or a year trying to collect. So this is always fun to look at, and this is really like a concrete piece of interpretable data to work with. So this is really kind of the fruits of all this data manipulation and processing. So this is really where you start to wear your biologist hat and think about what's happening in the system. Any questions on that? And then any questions? Okay, so the last thing I want to show you then is this last chunk of code. And what this is going to do is so here we took clusters 0 through 16 and calculate differentially expressed genes. And then we can use that to do annotation. However, we also have been provided annotation by the authors of the paper. So we can kind of cheat and skip to the end and be like, okay, here we already know the cell types. What are the genes that really label them? So that's what this last chunk of code does, is it just takes up the course cell annotations from the authors and it's too early in the morning. It reruns all that code that we just did. But it's going to do it for just the annotated cell subsets. So we'll just run that. So it's going to load up the course cell annotations. We're going to find all the markers, and then we're going to subset to just the top 10 genes, and we'll make a heat map again. So let's let that run in the background. And while that's running, any questions on differentially expressed genes? And right now we're running a very simple test, the Wilcox test. Like I said, it's doesn't assume anything about the distributions in your data. And it's generally decently fast to run and simple to understand. So that's why I like it. But yeah, this is running, it's saving. We'll open it up once it's saved. Okay, UMAP, DE genes, heat map, annotated cell subsets. Okay. Okay, so here are the subsets from the paper, from the authors of the paper: ciliate cells, goblet cells, secretory ionocytes, more ciliate cells, squamous, basal, and T cells. So now we can look and see, what genes define ciliated cells. And, if I knew anything about ciliated cells, I could tell you about what's going on here, but this is not my thing that I study. On goblet cells, I know a little more about because they're found in the gut as well, all the different kind of goblet cells. Ionocytes are a pretty cool population of cells. They were recently discovered, in the last 5 years. Notably, they have this gene CFTR, which you might have heard about in the context of cystic fibrosis. What else do we have? We have more we have squamous cells, which again, I know frighteningly little about. Do we have any cells that I know about? I know about T cells. So, so for T cells, we have all these different immune genes like CCL5 and CD69 and IL7R and granulysin and all those. So those make sense to me personally. Okay, but yeah, you can kind of see, like, depending on how you subset the data, you'll be studying different differentially expressed genes. And so often what we do is an iteration between clustering, like we might cluster in different ways and get different cluster assignments and then look at the differentially expressed genes. And we might say, aha, like these are T cells, but these genes here make no sense to me. Maybe we need to cluster it more or less or try something else. So there's often an iteration between this biological step where we're trying to interpret what these cell subsets are. And if we're confused or we think something's missing, we go back a step to the clustering or even earlier and change something and rerun through it again and look at how that changes, the genes that are being highlighted. So it's not, strictly speaking, single cell RNA sequencing has come a long way, the analysis, and there are far fewer choices I think you have to make than you had to make like 5 years ago. There are things you can just run and they'll probably be interpretable, but there's still this element of interpretation and cycling back. And so it's not quite a pipeline versus if you've ever done bulk RNA sequencing, that is basically a pipeline and that you almost never have to like iterate. But single cell, it's pretty common to kind of look and go back and kind of tweak things or try really different things. So this is why it's also much more fun and interesting and exciting. There's a lot of just like, using both computer science knowledge, but also biology knowledge to make meaning of this. So any other questions on differentially expressed genes or the clustering or what we've been discussing here? Okay, so I'll just end with, if we had more people, we have these little group exercises. We're not going to go through them because I think it's just too much talking through things and it's probably enough information already for one day. But I'll just tell you at a very broad level what they are. So if any of them are interest to you, you can just run them tomorrow or later today or in a week from now. And to run them, all you have to do is just hit that green arrow. Like there's no code that you need to write. So the first task was just interpreting PCA analysis and trying to choose like how many principal components you should put in your analysis. So that's an important question. And I think Isabella or somebody else might have asked how many to use. So that's, this task will give you a lot of different plots that will give you a more of a heuristic understanding of what the principal components are, what genes are important for each principal component, and where the signal is in the principal components and how many you need to include. So this is an important kind of technical question that often comes up. Second task is just asking, how do different parameters affect, how do different choices in the parameters affect like what you get from clustering and visualization? So, you can play around with some of these parameters for your nearest neighbor graph and the find neighbors. You can change this K value from anywhere from 2 to 100. You can also, instead of running a UMAP, you can run a TSNE and you can see how it's different. So again, just interesting to compare and give you more intuition as to how all these parameters affect your outcomes and how different visualizations might emphasize one difference better than another difference. Okay. And this next section is very important. It's about batch effects. So a batch effect are usually technical effects that we kind of want to know about and control for. So it might be that I do an experiment and Sergio does an experiment, and we try to combine our data sets. And when we plot it out, here's Orr's data set and here's Sergio's data set, and they're just not overlapping, even though we measured, say, the same thing. Like maybe we were both collecting airway tissue that had been infected with SARS-CoV-2, but we see no overlap. So that's commonly considered like a batch effect, where you've measured something biologically similar, but because of something technical, like maybe I'm not as good at doing the experiment as Sergio is, my data is systematically lower quality, and it just separates out because that's the most variation, is the fact that the data is just lower quality. So it's important to look for these batch effects. Other types of batch effects could be like, the experiment was done on separate days, or different labs collected the data, or there's like young and old people. That's more of a biological effect, but it might separate your data out in a way you don't expect. Or maybe there's sex specific differences, like maybe males are more affected than females. And so all your biological signal is really in tissue from males. Like there's all these things that could be happening that create variation in your data. And some of it you might not really care about or you want to remove. So that's what batch effect is about. And there's all these methods to do batch effect correction. So this section here just has you visually explore some of the data to see if there's any batch effects. So, we can look for cell types or states that are found within a single individual and not across the whole group. We can look at batch effects due to the sex of study individuals, or, if there's any systematic difference between the disease and control patients, and so forth. So that's an important one. And then finally, the last sort of little group thing here is asking what are the gene expression programs that are specific to COVID-19 patients. And this is conceptually simple in the sense that, we're going to choose a cell subset, like ciliated cells, and just do differential expression, where within ciliated cells, we compare the cells isolated from COVID-19 patients to cells isolated from the healthy controls. And we just do differential expression analysis there. And it doesn't have to be ciliated cells. It could be T cells or squamous cells or whatever cell type you care about that's in the data. And so that's going to tell you, what programs are induced potentially by SARS-CoV-2 infection. And so that's a really specific kind of biological question that's very interesting. And the programs might be different from one cell type to another that are induced. Okay. So that's sort of the little group things. And like I said, you can just run the green arrow and you'll see the outputs and you can play around with it. So I encourage you, once you feel like you have a handle on the main sections of the script, if you're still curious, to run all of them or just the one that interests you and, see what happens. So, yeah, so that's the lab. Any more questions people have? The questions have been really good so far, so happy to take a few more.
And just as a reminder to everyone here, you're welcome to continue to ask questions via the Slack channel that we've set up. So our training leaders have graciously agreed to check there and answer questions and help you out should you come across questions in the next week or so, or even further out. So
yeah, exactly. Thanks, Christine, for reminding them. And yeah, we get like email notifications when there's Slack messages, so feel free to use that. And then also, I think beyond that, the HCA Slack channels are very useful, and it's a really good resource for discussing some of these some of the things that come up as you become more advanced in your kind of understanding of these methods. I think also, let me just stop sharing. I'll also post, how do I stop sharing? I'll also post in the Slack a set of resources. I forget if it's was in one of the lectures already, but I'll just post it in the Slack. But it's a listing of, I'll share my screen in 1 second. Let me just find it. I think this is it. I forget if it was in the first lecture from Daniela or not. I probably was, but I'll repost it. So we have a lot more resources available to you that I think are really good. 1 second, it's just my computer's a little funky. Here we go. Okay, let me go back to sharing. So I'll post this in the Slack right now, but basically I'll just give you the bird's eye view. So this first set of links are listing of computational tools that people use in single cell RNA sequencing analysis. That's kind of nice to have it all in one place. Then we have these packages that automate a lot of the most common functionalities you need to do single cell analysis. So the most famous ones are Seurat, which we've been using in this course or workshop. But then if you're a Python user, all of this is also implemented in ScanPy. There are other workflows in R. The main one is the single cell bioconductor packages. And then there's another nice set of tools in Python that are a little more advanced called scverse. And this is really nice, especially when you start getting like multi-omic data, like gene expression and chromatin accessibility or things like that. These are a little more advanced tools. I think Dominic and Colin shared lots of articles on the HCA, but here's links to some and more of them. These links are very good here. These are online courses. I think they're all in R. I don't know if any are in Python, but you can click and find out. But these are all really good. I think one of them even is spatial transcriptomics. I think it's this one is spatial transcriptomics, which we're not discussing here, but you may have heard about it. It's doing these same types of analyses, but keeping the tissue coordinates and knowing where your cell was in space originally. And so you can get these nice spatial pictures of where the cells are in space and who they're next to and how they might be communicating with one another and what's the organization of the tissue. So that's another exciting topic, and you can learn about it or browse it in this book. And then finally, this is very, very not comprehensive, but these are single cell data repositories. 2 of them are from CZI, Tabula Muris and Tabula Sapiens, where they've done sort of whole animal organ and tissue collection across a mouse and across a human, and you can browse that data. There's the Human Cell Atlas, which is the biggest effort for cataloging human tissues in health. And then there's this cool website that you can go to where it's actually kind of crowdsourced, where it's a listing of all single cell data sets out there, or not all, but close. I don't even know close, but it's a lot. It's thousands and thousands and thousands. And there's useful metadata there, and you can search for things that you care about. And maybe this is a quick way to find existing data sets that might be useful to you. And it's crowdsourced, so like you can submit papers to it and it will be included in that repository. So I'm gonna just put this in the Slack so everybody has it, because I just forget if it's in lecture 1 or not, but I'll put that in the Slack. And so we'll take more questions, and then if there are no more questions, we'll I think just wrap up. But yeah, let's take a few. Any more questions people have, we're happy to answer. Just going to the Slack right now. I don't see any questions in the chat. Let's see. I'll just put this in the okay, sorry, it's just kind of like not the most beautifully formatted, but interpretable. So it's in, if you go to your Slack under your attendees November section, you'll see all the stuff we just posted or we just discussed. Okay, so let me give you guys 30 more seconds to come up with questions, and if not, we'll just give closing remarks. Let's see who's still here. Perfect. Okay. Okay, so I guess we'll close and we'll just say that we're really happy that you all showed up to this and are interested in doing single cell transcriptomics experimentally and computationally, and it's it's a really exciting time for the field. And there's a lot to be learned still, even though there's a lot of papers out there. There's so many biological questions that we think you can address with these methods. So super encouraging of you to keep down this path. And it takes time to learn these things, so be patient with yourselves. And there's a lot of resources out there, and everybody starts from not knowing any of this, so you're kind of in the same boat as everybody else has been. So keep at it, and we're really happy that you were all able to come to this and hopefully learn something. And then also, there's not too many people left, but please, Christine will remind you, please give us feedback in the survey so we can learn what worked and what didn't work, and how to improve this for future people in HCA Africa and these other training workshops. And yeah, so it's been really nice getting to know a few of you, and I hope everything goes well as you kind of go down this path. And then I'll hand it over to Collins and oh, Daniela's here, great, and Vincent, and if any of them want to say anything, I'll hand it over.
I just wanted to reiterate, it was great to work with everyone. Thanks for being here. So I just wanted to also echo my thank yous.
Yes, so I also want to thank this amazing team. They put a tremendous amount of time and effort into designing the curriculum. And I know you are all grateful to them. So just showing you everyone's names and faces one more time. So please do thank them with me here as we speak. I also want to bring to your attention that we're happy to announce that we will have our first in-person Africa workshop. It will be 2 tracks, one for computational folks and the other for experimental design folks. Depending on the track that you select, should you be selected, there will be some crossover techniques that you'll learn. So if you are interested in taking part in this workshop, please do register your interest here. I will, I can drop this in the chat really quickly. And just to let you know, this workshop will take place in Ghana, March 7th through the 10th, 2023. And please also share it with other folks who may be interested in partaking in this workshop. You'll see many of the training leaders from today's workshop in person in Ghana leading some of the trainings. The other thing that I want to mention, if you have not already registered to become a project registry member of HCA, we encourage you to do that. That's one of the best ways to join up with communities and to be informed of all of the seminars and trainings and meetings that we have throughout the course of the year. So please join up. Anyone is able, and it's free. And with that, like Orr said, please do share your feedback. We go through it, and all feedback is welcome, whether it be good, bad, or somewhere in between. And anyone else want to say anything before we leave? All right, well, thank you, and hope to see you at a future HCA event. Bye, everyone.
Thank you.
Thank you.